Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other for solving the problem. Here today we will discuss about the electron transport chain in mitochondria. So the electron transport chain are present in also in chloroplast, but here we will discuss about just mitochondria. So first of all we will discuss about the overview of the mitochondria and their structure and where these form. So the number of mitochondria present in the cell is here. So the number is variable because the activity will be induced to produce more. So mitochondria contain its own DNA and is able to replicate and divide. So the one mitochondria divide into two mitochondria in the same cell. But here is the we will discuss about the cell which contain an endoplasmic reticulum which is surrounded by the nucleus, and here is the Golgi bodies. That Golgi body is also used for the protein synthesis, protein maturation, and mitochondria are here, and peroxisome and cell membrane, plasma membrane, as well as cytosol or cytoplasm. So that's all thing is here, but no, here we will discuss about the mitochondria. So the mitochondria is contain double membrane bounded organelle. What is the meaning of double membrane bounded organelle? So the double membrane bounded organelle means that two bilayer of phospholipids are present. So here the inside is the mitochondrial membrane is known as the inner matrix of mitochondria, which is surrounded by the inner membrane of mitochondria. But the outer membrane is known as the outer membrane of mitochondria. So here is the F1 particle is the knob-like structure is also present, which that is used for the allow to move inside the pyruvate or some other use for other uses. So here is the inner matrix of mitochondria. So first of all we will discuss about the cross section of the mitochondria with the middle. So if we will discuss that the cross section in the middle side, so the mitochondria is look like the structure is look like the crystal structure. So there's crystal formation is due to the invagination of inner mitochondrial membrane. So here is the cross section which that shows that the Krebs cycle are present inside, while the outer mitochondrial matrix is also allowed to move inside. So the inner and outer mitochondrial matrix is surrounded by the inner and outer mitochondrial membrane. So is the crystal formation. So electron transport chain is occur inside of the mitochondria. So the mitochondria is produce ATP as well as carbon dioxide by the respiration. So the cellular respiration is occur due to the mitochondria, and it is also known as the powerhouse of the cell. Powerhouse of the cell mean which produce the energy. So the major source of production of energy is the mitochondria. But on the other hand, there is the cytoplasm also produce energy in the form of glycolysis. That's glycolysis mean the glucose formation, glucose form to convert pyruvate, and that's produce NADH and ATP, but less amount of NADH and ATP. So the pyruvate is further go inside of the mitochondria to convert the acetyl-CoA, and this acetyl-CoA will convert with the mitochondria. So here is the another diagram which that is used for understanding that the inner and outer mitochondrial matrix, which contain inner mitochondrial membrane contain electron transport chain. So there are four complexes, five complexes are present in the inside of the mitochondrial membrane. So there are many complexes are present, but we just for understanding we will consider the five mitochondrial protein subunit is known as the complexes for understanding, but there are many more complexes are present which type of 5 type of complexes, but the type is 5. On the other hand, there are many more. So let's talk about the electron transport chain to understand the better way, and that I will try to make a diagram with a good manner for understanding just. So the simplicity I want to create for understanding, so that's why I decided to make a new video about the electron transport chain to understand with a simply. So on the other hand, the mitochondria increase due to the increase of the exercise. So the exercise increase the number of mitochondria due to more ATP required. So the more active to produce energy and require more energy and heat. So that's why mitochondria will be divided, and this division will allow to more sugar consumption in the form of pyruvate, and that's pyruvate produce the acetyl-CoA to produce ATP. So that's a lot of ATP will be produced when we will running state. So after the running, when we will sleep, that's a mitochondrial division will be occur. So I hope you make sense about the mitochondria, which that is a powerhouse of the energy which produce the ATP, and this ATP is used as a energy for different cellular function, and even every function or every physiology of the body is use ATP to produce energy or development. So here I am drawing the electron transport chain which contain complexes. So here is the outer mitochondrial membrane, and inner mitochondrial membrane is the inside. So the outer is the outer and inner is the inside, which that is in the form of cristae, which that is a valve-like structure are present. So inner side contain a complexes. So these complexes is, for example, capsular-like membrane which surround whole mitochondria, and these is many more complexes are present. But here we will just draw 4 complexes for understanding. So the 4 complexes is inner mitochondrial matrix contain a Krebs cycle. So the Krebs cycle is begin inside of the mitochondrial matrix by the fixation of acetyl-CoA, and acetyl-CoA will come from the beta oxidation of fat or other. So here we will discuss about the complexes. So the complex is one which that is known as also NADH dehydrogenase, because it's the enzymatical activity. So on the other hand, the complex 2, which that is also known as the succinate dehydrogenase. It means the succinate conversion into the fumarate. That is why it's known as the succinate dehydrogenase, which that is a Krebs cycle produce succinate, and fumarate will be produced by the Krebs cycle. So that's character. And complex 3 made up of cytochrome C oxireductase. It means oxireductase mean oxidation and reduction in the same complexes will be occur. And complex 4 is known as the cytochrome C oxidase, which that is used for the oxidation. And the last one is also known as the ATP synthetase. We will discuss in this video. So here I am drawing that is a fatty acid which is contain a carboxylic group, and this fatty acid is the long chain of fatty acid. We go inside of the inner mitochondrial matrix and convert by the beta oxidation to produce acetyl-CoA. So it means there are many more acetyl-CoA will be fine. This beta oxidation is the produce acetyl-CoA many more. One beta oxidation to produce one acetyl-CoA. That will go into the Krebs cycle and will bind with the Krebs cycle, that is the acetyl-CoA. And that means acetyl-CoA is not use the oxygen, but the acetyl-CoA will be enter inside to produce oxaloacetate into the citrate. And this oxaloacetate convert into the citrate, and citrate convert alpha-ketoglutarate and succinyl-CoA conversion, and that succinate to produce a basically fumarate to malate, and that's malate dehydrogenase to produce again oxaloacetate. So these six carbon containing compound is used as a production of energy, NADH and FADH2, as well as ATP, 1 ATP and the NADH, 6 NADH with a 2 turn due to the 1 glucose for 1 glucose breakdown to produce 2 pyruvate. So that's why. So here's the oxaloacetate will be formed from the malate, and that's produced in this reaction to malate dehydrogenase enzyme to produce NADH. And these basically we are understanding the Krebs cycle to understand the electron transport chain. So here is the NADH plus convert into the NADPH. So the reductase enzyme, which that is basically succinase enzyme used to I don't know about that enzyme, but remember that is enzyme used for it to make 2 NADH from 2 turn, and two basically 3 carbon dioxide will be formed from 1 turn. So that means ATP is also produced and CoA will be released, and this NADH will go into the complex 1 NADH dehydrogenase, which that is used for the dehydrogenation of NADH to convert the basically to convert the NAD plus. So this production will be again formed, this cycle will begin from here, again Krebs cycle will be capture the NAD plus. So this is the here is the FMN is a protein which that is pick these 2 electron, and 2 proton will be oxidized outside of the inner mitochondrial matrix. So the 4 proton will be move upward due to the cyclic activity of the iron sulfur containing protein to move upward the 4 proton. But here is the 2 electron which that is pick the co and plus to basically coenzyme Q, which that is used to reduce in the form of H2. So these 2 proton will be picked from the inner mitochondrial matrix, and that means the convert into the CoQH2, and this CoQH2 is reduced by the 2 electron and 2 proton, and in this case this CoQH2 will be go into the third complex is known as cytochrome C oxyreductase. So it means it's go into the third, not second. So the second you are missing. So we are missing for understanding or another mechanism for understanding. So it is not involved in this mechanism. So CoQH2 is oxidized by the oxidation, that is the oxidase enzyme used to convert CoQ, which that is again give the iron sulfur protein which that contain a complex 3, and this iron sulfur protein accept this electron, but 2 proton will be 2 proton will form inside of the matrix to move outside by the pumping due to the oxidation reduction mechanism. So these 4 proton will be formed because H2 coenzyme Q, which contain H2, and it means its release. It's known as the reduction oxidation. So these 4 proton will be formed by collectively. So this rotation will be formed, and this rotation will pick that proton by the using of electron energy, which contain electron energy. So the electron move forward to decrease the energy, and this energy decrease due to the proton pump outside. So the outside proton pump outside will be formed. So here is the 4 proton due to the CoQH2 is 2 proton. On the other hand, 2 proton is pumped from the inner mitochondrial matrix to the outer mitochondrial matrix. So here is the cytochrome C, that is cytochrome C contain, again that is the reduction. So the cytochrome C reduced in the form of cytochrome C2 electron. So this pick the 2 electron is known as the reductase enzyme reduction. So the reductase enzyme used to reduce in the form of cytochrome C, with 2 electron. So that here is the cytochrome C. So these cytochrome Cs go into the next complex, is the complex 4, is also known as cytochrome C oxidase. It means the oxidize the cytochrome C. So oxidize means the releasing of electron. So the releasing of electron move back the cytochrome C. So the cytochrome C is here, and this cytochrome C is backward by the oxidation to again pick the electron from the complex 3, cytochrome oxyreductase, from cytochrome oxyreductase. So here is the complex 4, which that contain a cytochrome C oxidase. So the cytochrome C oxidase is contain a 3 subunit of iron sulfur protein is here also, and 1 2 3, and these contain a rotation. So this rotation is give the for example proton will pick inside. So you can see that here is the one proton will be released inside, and that's proton bind where electron bind where that's a 2 proton will be attached. So it means basically it's contain a basically it's contain a 6 proton. But on the other hand, when one more proton will become, one 2 electron will become, it means the 2 electron will be add more proton from the inner mitochondrial matrix to the for example complex 1, and that's bind to extra which that was the previous 2 proton which release outside. So that is why it is take the place for itself. So the 2 electron will be combined to the proton, and this proton will be 2 electron lose the energy to pick up the proton from the inner mitochondrial matrix to fix inside of the complex 1. So the fixing of the complex 1, the 2 proton will be left behind because 2 proton was already, so that's go outside. It's known as the oxidation. So after all, electron will lose energy, and after all that losing of the energy, electron is unable to move forward. If it able to move forward, it can carry the another complex again. So another complex is pick or accept is very difficult. So here the 2 proton will be enter. So the 2 proton will convert pick the 2 electron. So the 2 electron will pick, and here is the half molecule of oxygen will bind with the 2 proton and 2 electron to form water. So it means the electron which flowing of electron will be clear here, but very important to understand that the 1 FADH2 is produced 6 proton. It means that in this structure or in this illustration, the one complex 1 will be pump out the 4 proton, and complex 2 will not pump, and complex 3 will pump out the 4 proton more, and complex 4 will pump out the 2 proton. So the proton basically, so that's 4 and 4 is 8, and 2 more is equal to 10 proton. So the 1 NADH produce the 10 proton. It means the 4 proton used to produce 1 ATP. So it means the 2.5 ATP will be produced from NADH. So here we will discuss about the FADH2 is also produced from the Krebs cycle. So the FADH2 is also produced from the Krebs cycle. So here is, you can say that the succinate convert into the fumarate by the using of enzyme is known as succinate dehydrogenase enzyme. and this succinate dehydrogenase enzyme used to produce FAD plus to convert FADH2. And this enzyme here is the important enzyme here next we will discuss about here, but you should understand that glycerol 3 phosphate dehydrogenase or flavoprotein dehydrogenase enzyme 2 which that are present is here. So next we will discuss, but first of all we will discuss about the FAD plus conversion into the FADH2. So the succinate dehydrogenase enzyme is used during the Krebs cycle to produce FADH2 from the succinate dehydrogenation to form a fumarate. So the two molecule of two proton will be loose, and these two proton will be bind with the FAD plus to convert the FADH2. So this FADH2 is picked by the CoQ. So here is the FADH2 will yield the ATP is due to the 4 proton is produced from the complex 3 and complex 4 produce 2. It means the 6, it means the 1.5 ATP will be produced. So on the other hand, glycolysis is also present. It means it's not inside of the mitochondria. So that is in the cytoplasm. So the cytoplasm contain glycolysis. So the glycolysis step to produce 2-pyruvate, and in the result of that yield the 2-NADH. So the 2-NADH is outside of the outside of mitochondria, which that use a mechanism is known as the glycerol 3 phosphate dehydrogenase enzyme 1. So the glycerol 3 phosphate shuttle mechanism is used to inside movement of this proton. So the NADH is the energy unit, so which that is a precursor of ATP. Remember about that. So the glycerol 3 phosphate dehydrogenase enzyme 1 will be formed and pick the proton by the dehydrogenation to give the dihydroxyacetone to the with the phosphate group attachment. So this phosphate group attachment is here. That is the dihydroxyacetone is here, which contain a 3 carbon containing molecule which bind with the phosphate group with the help of the 2 proton. And these 2 proton will be bind to form a glycerol 3 phosphate. And this glycerol 3 phosphate move inside of the complex 2 succinate dehydrogenase, and these move inside to produce FADH2 to convert FAD plus. So FADH2 to convert FAD plus. So the FAD plus it's FAD plus convert into the FADH2. So this give the 2 proton by the using of FADH2 to give the CoQ, and this CoQ to capture and this oxidation to release the 4 proton. Same mechanism. So it means 4 proton is come from the complex 3 and 2 will be here. So it means the 6. It means the 2 NADH from the glycolysis is produce the 3 ATP. So it means 1.5 ATP is also produced from the glycolysis. So the 2 NADH will convert into the 3 ATPs also. From this mechanism is known as glycerol 3 phosphate shuttle mechanism. So here that is the 5 complex is also known as ATP synthetase, which that is here is the outside the proton concentration is too much due to the pumping and produce the concentration gradient, and after this stored in the form of energy, and this energy will be stored in the outer mitochondrial matrix and will not allow to move proton inside. So anything is not useful except the ATP synthetase. So the here is a go inside, and here is the rotator, and that is the stator is also which that fix the that is a machinery which they are used to enter the proton 1 and 1 release from the there are many subunit, beta, epsilon, delta, gamma, as well as F1, no, F1 region and F naught portion of this part. So here is the movement of the proton from here, and that is moved from the beta region to move inside, and in this process the beta region of the beta subunit of protein, which that is used for the ADP to convert ATP. On the other hand, that's movement. First of all, we will discuss about the movement. So here is the B2 presence rod-like structure of the subunit of the protein, and here is the alpha, and this alpha and B, and that is many more. So here we will not discuss about that things. So here the rotation will be formed the proton fill. So this proton filling of this proton, one proton move inside and one proton will be enter one more out from outside. So this rotation one by one proton move inside to produce ATP by the using a 4 proton. So the 4 proton used to give the phosphate group to give the energy to bind with the ADP to convert ATP. So that is the mechanism for using as a energy. So important thing is that is why that is the concentration gradient will be formed, and this concentration gradient is important to understand, and that is concentration gradient is used without energy to move outside to inside. So that is why proton contain energy, and that is why here is the DNA of the mitochondria. So remember about that thing. Mitochondria contain its own DNA. So it can stimulate the ATP to use bind with the DNA by the cyclic AMP, the second messenger, to release the protein to synthesis more complexes, for synthesis of more complexes. So that here is the ATP will be produced by this activity. So remember that the one proton will be released and the one fill again, and the rotation will be again and again. The movement upper part is the rotator F naught. On the other hand, the F1 is the stator, which that live in the state. So that is epsilon unit of that, which that's control to prevention of rotation. So here the inside of the proton concentration is less, so the proton moves inside again to level the proton concentration. So important to note that is the concentration gradient will produce, and this concentration gradient is allowed to move inside of a proton to, that is the proton energy is used as a storage energy in the form of ATP. So that is a simple science to understand these things. So here is the I am drawing the table for understanding and calculation of the ATP by the glycerol 3 phosphate glycerol 3 phosphate dehydrogenase enzyme to use to the glycerol 3 phosphate shuttle mechanism to produce. Shuttle mechanism is why, because here is the shuttle 2 enzyme, inside is 1 is same like the outer mitochondrial membrane, which that is pick the electron, and that's glycerol 3 phosphate again convert into the dihydroxyacetone, and these 2 proton will be can give to the FAD plus to convert FADH2, or give to the coenzyme Q, which that's use and that's peak. So here is the 1 FADH to produce 6 proton is equal to 1.5 ATP, and glycolysis produce 2 NADH, which that is equal to 6 proton multiply, and the scrap cycle produce 3 NADH with the 1 turn to produce 3 into 10 proton, because it's not any other mechanism. So simple on the basis of different mechanism. So here we will discuss about the reaction step. So here is the glycolysis, which produce 2 ATP and 2 is used. On the other hand, 2 NADH will be produced. Here is the Krebs cycle, which produce 2 ATP with the 2 turn. On the other hand, 3 NADH with the single turn, and the single turn with the contain and 1 FADH2. But here I am again writing that the Krebs cycle in 2 turn. So the 2 turn are double turn to produce 2 ATP, same here, but 6 NADH will be fine, and that's 6 NADH and 2 FADH2 is FADH2 will be formed, and here is the total ATP will be produced from the this mechanism without electron transport chain is here 6. So that is mean here is a 6 ATP will be produced by this mechanism except the electron transport chain. But when go into the electron transport chain, for example, when go into the electron transport chain, so NADH will be used and FADH2 will be used. So the NADH and FADH2 will be used how it's produce energy. So its total is 11 NADH and 2 FADH2. So this, if we will talk about the multiplication, so here we will discuss and multiply 6 into 2 is equal to 12 our 4 mean 4 proton which produce 1 ATP. So here is the basic glycolysis is due to the glycerol 3 phosphate shuttle mechanism to produce 3 ATP by the 4. So here is the 3 into 10 to produce 30 our 4 because 3 into 10 because it is the electron transport chain of the Krebs cycle to which that is the Krebs cycle produce NADH to produce 7.5 ATP. So the here is the 3 into 10 to basically here is the further 30 to 30 over 4 to double turn. It means the 2 turn of the Krebs cycle to produce 15 ATP. But here is the 6 ATP is also produced. So that this 6 ATP is here, and this 6 ATP is basically 18 ATP is formed. So the 18 ATP is here, and this 18 and 6 ATP will form to produce 24 ATP yield with the 1 glucose molecule breakdown. So the first of all I want to say that the glycolysis is produce the total ATP is 4 ATP. So the 4 ATP means the 2 ATP will be used and left behind 2 ATP. That is the total using ATP, and the total NADPH is used to basically total NADH will be formed is the 2 NADH, and this net is the 2 NADH will be formed and 2 ATP. So that is the glycolysis, and to produce that ATP is given below. That is the basically 12 proton will be formed from the 2 NADH. On the other hand, the 20 proton will be formed in the Krebs cycle when NADH used from the Krebs cycle inside of the mitochondria. Okay, here we had understood about the proton concentration and proton by the different mechanism to understand the glycerol 3 phosphate dehydrogenase enzyme to use it, 1 and 2, which that is one is the outer mitochondrial membrane and one is the inner mitochondrial membrane, and on the other hand the complex 2 succinate dehydrogenase enzyme is not produce the proton. So that is why. But here in the textbook of the A level or O level, which gives the 36 ATP and 2 ATP will be used in the glycolysis and left is the 34 ATP. So the 34 ATP calculation by this mechanism it's not used for the understanding. So that is the simple system which that is the glycolysis produce 2 ATP 2 NADH and Krebs cycle produce 2 ATP is equal to 2, and it's here is the total NADH will be formed and total FADH will be formed, basically 2 FADH2 form, and that's the total will be 2 into 3 is equal to 6. So it means that is the 6 ATP will be formed, and that's here is the total 4 ATP is formed from the Krebs cycle glycolysis, and that's here is the 6 from here, and the 6 ATP is from here, and on the other hand 24 is come from the electron transport chain, and that is here is a 4 into, and that is convert into the 33. So 34. So 34 means here is the 2. So that is why here is the 2, and that is why here is the 2, and here is the my mistake about that. So we will add one more to finish. So here we had understood about the electron transport chain. So here is the done the video.